Next, Krishna, who has sorted out all the audio issues. Let me introduce Krishna real quick. Krishna is with Infinome Biosciences, and he has been with us in I think at least one other fireside chat about bioscience, biotech, and Flyte. And I think this was very interesting. July, end of July, a little bit ago. And we said, well, let's listen to how Infinome is using Flyte and get Krishna back here. So Krishna, please unmute yourself, introduce yourself, and tell us a little bit about your background, and then we're going to kick off the presentation in a second.
Awesome. Can you hear me?
Yeah.
Awesome. All right. Yeah, so thanks for that introduction. I realized that picture is several years old now. And I've grown a beard since then. Yeah, so I am a data scientist in Infinome. So my formal background is in chemical engineering. I did some machine learning as part of my dissertation, actually neural networks before deep learning was a thing. And then I worked in several biotech companies since then, mostly on data science, statistics, not necessarily on the data engineering side of things. So only in the past, maybe 1 year, maybe 1.5 years, I've been actually trying to work a little bit or know a little bit more about workflow management tools. And in my previous job, I was exposed a little bit to Prefect. And when I started at Infinome, I wanted to kind of stand up some of our analysis pipelines. And that's where we ran into Flyte and the community, and everything has been super helpful so far. And we actually have a couple of pipelines that are running on Flyte right now, and people have been using them. So
fantastic, love it. So to your presentation, what are you going to talk about? And do we hand it over to you, or do we have these slides on our side? I don't know.
Oh, I didn't send you the slides. I can share my screen and kick off on mine, and then I can share my slides after the presentation.
Fantastic.
Yep. Awesome. Cool.
Okay, cool. So, Krishna, tell us a little bit about Infinome. I don't know if many people know about the company, but how big is it, and what is Infinome doing?
Yeah, so Infinome right now is 18 people. So we're a synthetic biology company. We were founded in 2020, right in the middle of the pandemic. So what we're trying to do is what we call lean bioengineering. So synthetic biology, for people who are not familiar, is using microbes essentially to make products that are typically made using other chemical processes, typically, maybe let's say petroleum-based raw materials. So what we want to do is, start making synthetic biology profitable in some ways and faster. So part of that is what we're calling lean bioengineering, where instead of putting, let's say, months to actually make one improvement or one cycle of improvement, we want to reduce that time to weeks. And also, instead of putting tens of people on it, can we actually have just 2 or 3 people per project and still achieve improvements within weeks? So that's what we're calling lean bioengineering. And that typically has been the challenge with traditional synthetic biology companies, is getting those products off the ground. Even though I think there's a lot of demand right now from the market, it's harder for biology to work compared to chemistry. So
interesting. So when you joined the company, obviously shortly after you got funded. So I assume that you're the only data scientist on the team, or a small group of data scientists. And how did that go when the data scientist joins a team of I guess engineers?
Yeah, so they're biologists essentially. So so the CEO is a data scientist. But he's also a biologist in the sense that he's published a very seminal paper actually called ProSAR, which is using machine learning for protein engineering. So he kind of straddles, I guess, both worlds. So he's the first data scientist, I think, in the company, and then he hired me. So right now we're planning to hire one more person to do that. So the interactions with biology, I think one of the things that we learned, I think I'll highlight this in the slide as well, is we want to put biology first and make sure that the bench scientists' life is easy. We don't want them to jump through hoops to submit, let's say, a job, an NGS job. They don't have to worry about what columns they need to, exact formats of files and all those things. So we want to make their life as easy as possible. And that is why we wanted to choose something like Flyte, so that they can actually use these self-serving apps to submit jobs, look at their results, and make any tweaks to the parameters if needed, instead of data science being the middleman, because they have the best context for data rather than someone like me, especially when there's multiple projects going on at the same time. Awesome. So yeah, so if you go to the next slide, Sandra, so the agenda, like I said, is, I'll give you a brief introduction to Infinome, which probably we already did. And then I'll talk about some things that led me to choosing Flyte at Infinome. And then we'll talk about a couple of pipelines that we stood up in the past few weeks. And then the lessons learned at the end, and take any questions if you have any. So I think we have 40 more minutes, so we can go slow. Please stop me. And if I throw acronyms at you, please stop me. Yeah, so let's give this a discussion. Yeah. So like I said, my professional experience, I'm not a data engineer by any stretch. Only in the past 1 year, I've been dabbling into this stuff. So I'm a chemical engineer, mostly machine learning, statistics, working in biotech. And I'm part of a 2-man data science team along with our CEO. We're looking to hire 1 more member soon. So if you're interested in working in synthetic biology / data science, reach out to me. So the next, if you go to the next slide. Perfect. So Infinome, like I said, we were founded in 2020. We have 2 locations. One is in Boulder, Colorado, where we do a lot of our microbiology. And that's where our microbiology labs are. And the other location is in Burlingame, close to the airport in San Francisco or South San Francisco, where we have a lot of our analytics machines. So when I say analytics, it's GC, gas chromatography, mass spec. It's not the analysis that we typically do with number crunching. That's something that it took me a while to understand when people were talking about analytics. Is something different from analysis. Yeah, so that's where we're located, probably half and half personal between those 2 locations. And the leadership team has a lot of experience in biotechnology. They come from Codexis, other previous successful companies like Codexis, Emergen, Intrexon, Novozymes, and so on. And if you go to the next slide. So the problems, like we discussed before, with the traditional way of doing things is they're slow, and they need a lot of people working for each of these projects. So what we wanted to do is combine a couple of new technologies that came out in the recent decade or so, maybe less than that. One is called CRISPR, if you're aware of it. It's a technology that lets you make very precise changes in a genome. Like if it's a plant genome, it's a bacteria genome, whatever. And for that, these 2 ladies, Jennifer Doudna and Emmanuel Carpentier, won the Nobel Prize in 2020. So we want to combine that with what we call directed evolution, for which Frances Arnold, she won the Nobel Prize in 2018. So combining these 2 tools or techniques, we are able to speed up the cycles from, like I said, months to weeks, and also get away with having only 2 or 3 people working for project instead of having tens of people working per project, while driving the same improvements over time. Yeah, that's what we're calling lean bioengineering. If you go to the next slide. There's a lot to take in, but what I wanted to highlight is this is an iterative process. So typically we start with a single cell. There's nothing it's a common, let's say, E. coli cell, and we make single precise changes using CRISPR, and then we test these cells using different techniques like sequencing or mass spec or gas chromatography, and these machines generate a lot of data. And then we analyze these data and say, hey, this particular cell with this particular mutation seems to be performing better than the other cells. So once we know what cells are doing better than others, we can combine the mutations within those cells in the next round. So instead of having 1 mutation per cell, we can have 2 mutations per cell. And once we do this for 4 or 5 cycles, hopefully at the end, we have a cell that has 4 or 5 different mutations and is much, much, much better than what we started out with initially. Whether that's for producing, let's say insulin, for example, or for any other product. So that's the general cycle of how we do things. And
So
this
quick question here. So I mean, looking, this is pretty complex here, and obviously it's a small company, but this you have to have a lot of automation. Now, from your point of view, obviously you're sitting probably in the top right of the slide, right, at the analysis.
That's
right. And is that just to understand more classical, pair t-test, or comparison of independent groups and see what that means, how they differ, or can you tell us a little bit about what you're doing there?
Yeah, good question. So there are parts of workflows. There are different workflows that do kind of a very complicated t-test or pairwise comparisons, different conditions. A popular tool, if you know, is something called DESeq2. It's been there forever in RNA-seq world, where you compare different populations and say, this population has something different compared to the other population. That's pretty much what we do with one workflow. And the other workflow is more statistics than anything else, trying to understand, if you put these two things together, does the data tell us that they are synergistic? Right. And then we say this combination seems to be better than either of those by themselves, for example. So there's a little bit of statistics there. And yeah, so it's mainly been traditional. Once you get that raw data processed, it's statistics mostly. It doesn't even come into like what we call machine learning right now. But having said that, we have done a few things in the past where, with the recent advances in NLP for proteins, for example, we are trying to, I guess, reduce our world of things that we can test using these priors that we already have based on, let's say, an NLP model that says, if you put this amino acid next to this amino acid, maybe you'll get a protein that's not going to do as well. So if we have that information, we can reduce the space of things that we need to test, that we have to test. So that's something that we are also exploring, which is more, I guess, contemporary machine learning.
Yeah. And I think the interesting takeaway, at least for me, as looking at this with no prior knowledge about this, is that you can do classical statistical modeling and t-tests and hypothesis testing and use workflows that are built on Flyte to really actually make sure you have reproducibility. It's not always that you have to go all the way to building complex machine learning models. Is that correct?
Yes. So right now, what we're using Flyte for is to take the data coming off these machines and make it in a way, or process it in a way that we can actually do these t-tests. So we're not even using Flyte for t-tests yet. That is something that we want to do going forward, is make that standardized as well. But for now, the things that are easy to standardize and give us the most benefit are the pre-processing steps between the machine and the t-test, if that makes sense. Yeah.
All right, thanks.
Yeah, but the value obviously comes from those final tests or analysis where you actually get something actionable out of them. Yeah. So if you can go to the next slide. Any more questions, by the way, so far? I know I spewed a lot of biology and acronyms at you, so. All right, so this is where the rubber meets the road, I guess, for this presentation. So what are the challenges we are facing? For us, I think there's a good difference that I kind of started appreciating in the past couple of years with typical companies that use workflow management tools or even software pipelines versus most biology companies, is the analysis pipelines are evolving really fast. So there's no pipeline that you can build today and expect to use for 1 year without making major changes. That doesn't happen in biology. Things change so fast because we're still in the research phase, right? So we need to keep up with those evolving pipelines, and we don't want to spend too much time developing pipelines and standing them up and trying to do, really make sure that everything is fail-safe. Failures are okay because we can deal with them. If it's 1 in 10, that's fine. So that's the major challenge. And then we have a hierarchy of analysis. So like I said, the pre-processing step, taking the data from the machines, making it a little bit more human readable is the maximum value, I think, for now. That's what we are building the pipelines for Flyte, using Flyte for right now. The secondary is what Martin was talking about, t-tests, enrichment, depletion. Those are secondary analysis. We want to get there, hopefully, by the end of this year. And the tertiary analysis is a little bit more complicated, but I don't know if there are any common threads that we can use Flyte for. We still have to ruminate on that internally. But the key thing I think for us is both for biology and the data workflows, we want to identify common patterns across multiple projects because we're dealing with more than 6 projects right now. And I can't keep up with all of them, what's happening with all of them. So we want to make sure that most of these things are automated as much as possible. So as we go along this journey, because we're a small company, we're still learning with these different projects right now, we want to identify what worked for biology, what worked for analysis, and automate each of those separately. And probably Flyte is the way to go for automating some of these workflows. And the last thing I want to say is biology is always the bottleneck. It's hard. So we want to make life easier for bench scientists. And even if that means sometimes custom pipelines need to be built, or we need to spend a lot of time trying to make a particular file format easily readable by biologists, that's definitely what we want to do. We don't want to take the shortcuts for data science and make life easy for biology. And that's where I think Flyte really shines for us. If you go to the next slide. So why Flyte? So like I said, we want to give the power back to biologists. So we want to stand up something that they can actually use, play around with different parameters for their models, because not every parameter is fixed. So with some of these workflows, there's nuances to each data set. So we want to make sure that we are giving them the power to run these analysis and not having data science be an intermediate for whatever reason, like because it results in slow turnover of analysis. And I've seen that multiple times in my career. So that's where we wanted to use something like a workflow manager like Flyte. Can you still hear me, by the way? Yeah, cool. And the other thing is most of these bioinformatics pipelines are kind of like data flow programming models, right? Like you push data through several of these small tools. And again, having tasks and Flyte, how Flyte treats tasks is perfect for what we want to do. Maybe there's a better way of doing this, but for me, I've always thought about, tasks as the key for most of our bioinformatics pipelines. And some of these tasks are trivial and fast, while others are slow. And also dynamic workflows are really important. And we just don't want to use all static workflows all the time because that becomes really hard to program. And be able to work with Python, R, and hopefully Julia too in the future. And having container tasks, at least on paper, seems to be the way to go, even though I haven't really personally tested. So far, we've only been using Python, PyFlyte. But hopefully in the future, we'll start using R a little bit as well.
Yeah. So one thing that comes to mind, and I probably see that the data passes through lots of processing steps. What does that look like when you don't use an orchestrator like Flyte? I mean, what's that world? Tell us a little bit about that experience.
Yeah, it's even before. So I use Nextflow. We'll talk about it a little bit briefly, but even before that, lots of people use Bash scripts, right? So they just put these things together. I've done that in the past. So and it's not tractable. You don't know where things are failing and you don't have any reproducibility for some of these things. So yeah, so that's bioinformatics is still a wild west in that regard. So
Right. Okay, fantastic. Yeah, I'm aware of some of those processes. And then being able to work with Python R, I mean, you can work with any language R as well in a raw format that's supported today on Flyte. But obviously there's a missing SDK, but maybe that's something for the quarterly planning that we can talk about that topic for Flyte. That might be a good topic. Okay, fantastic. Let's keep going.
Yeah, good question. Any more questions so far? Again, apologies if I don't use the correct language because I'm not a data engineer, but please feel free to ask any questions or comments.
Hey, Krishna, this is Ketan. I'm from Union. I have a question for you, and this is probably orthogonal to the current part of your talk, but like you said, you have currently using Python SDK. How many use cases do you think are in our Julia? And the reason why I'm asking this is because across multiple recent talks, we have heard R a few times. From a community point of view, I'm just trying to gauge what is the interest as well as needed to have an R SDK, a native support for R within Flyte.
Yeah, I think I mean, over the years, I think Python has actually been more widely used than R over time. So the trend is, I think, increasing towards Python over time that I've seen, even within biotech. But R still has a market, definitely in statistics. So you can beat R with some of the libraries that academics, they push out a lot of R libraries consistently, and Python statistics libraries are still a little bit lagging behind R. So that is I think, the definite use case for R is some of these libraries. And I can send you a few that are used over and over in bioinformatics. I mean, if you can even search single cell pipelines in R. There's so many of them. Some of them are starting to be developed in Python as well, but R, I think, is still the main NGS workflow tool or library.
That is fantastic to know. This is what I wanted to know about, like, it's basically within, so it's correct to say that it's within the domain of biotech and bioengineering. I think R is dominant for many use cases still.
That's right.
And a native R integration might really accelerate your workflows or the way you develop it. Right?
Yeah.
Make your life easier, essentially.
Exactly. Yeah, I think so. Yeah, it'll, if that is part of Flyte, I think biotech is almost, I don't want to say that's pretty much 80%, I think, of what people in biotech use. Python plus R should cover most of it.
Yeah, and if I just add to that, obviously, with I mean, on the statistical modeling side, it makes a lot of sense. Most people are using R there, and also not only in bio but in life sciences, when you have things like FDA submissions and so on as well. I think that's a whole other area where there was another tool. It was SAS that was dominant, that has changed, right? There's a change from SAS to R and on the science side that is, I think, very noticeable. And I feel like on anything that is ML related, it's not really the domain of R, even so you can do it, but people know that there are better tools, and that's all in Python. So I feel like data scientists today have to do at least 2 languages, and in many cases actually Julia is really an important part. You cannot just be just saying with one language I'm going to be good. I think those days are over. I don't know. Do you agree with that statement?
Yeah, I think the SAS to R, I think I've seen that in the past. I haven't kept up with it recently, but that's definitely true. Lots of people have transitioned over to R from SAS. All those traditional, I think you're right, with the FDA, old school statistical analysis companies did transition over to R. Yeah, so there's that market. Yeah.
All right, thanks.
Yeah. So if you go to the next slide. So yeah, so I guess 4 different next generation workflows are probably common within Infinome. So edit detection, this is the main workflow that wanted me to go the route of Kubernetes and Flyte, because it was, at that time, it was really slow. It took 5 days, 6 days to run for 100 samples. So we wanted something that scales really well with samples and is super fast. So that was the perfect use. I was exploring Kubernetes, and then I ran into Flyte, and I thought, oh, this is perfect marriage. So that's how we implemented it. And now it's currently, I think, less than a few hours, depending on the number of samples, we can get the results back. The second one that we stood up recently is Plasmid QC, which has some unique challenges in the sense that it uses a lot of different tools, and also there's some scaling issues depending on the number of samples and read depth. The third one, which we haven't implemented yet, which is a mix of R and Python to do the t-test comparison stuff that Martin was talking about. And the last one is more traditional or contemporary ML workflows that we have done in the past, but we want to resurrect in Infinome. Hopefully, in the past, we used SageMaker, and I've seen some cool things that you guys are doing with Flyte and SageMaker, so I'm excited at some point to test it out as well. So. Cool. And if you go to the next slide, I'll show you a couple of So what we've done is we kind of combined Flyte with Streamlit. So we've used Streamlit in the past, and like I said, because our analysis workflows change so often, and even our data ingestion changes so often, we don't want to go down the route of trying to like Dash or something, because yeah, I mean, Dash has some advantages, but Streamlit, because it's Python based and it's so much easier to just have a slider using one line of Python code. I love Streamlit. So I combined Flyte with Streamlit. So using Streamlit, biologists can submit their jobs. They can choose the parameters, for example, using a drop-down menu and say, hey, these are my parameters for this job. This is where you can find the data on S3. Just run this pipeline for me. And then they can see the results in Streamlit again. And then, yeah, so like I said, it takes a few hours right now compared to a few days in the past. One thing with this particular pipeline is there's a lot of small map tasks for which I think the Ray integration is probably the right thing to do. I don't know anything about Ray, but it seems like, at least based on some Google searches, that there might be some efficiency that we can get out of it by using that instead of simple map tasks. So this was our first pipeline, works beautifully so far. People have been using it with some minor issues here and there, but nothing to do with the app itself. And if you go to the next slide, this was the most recent one. Again, the same philosophy: Streamlit as the front end, Flyte as the back end to run these jobs. This is a complex pipeline with multiple tools. So there's a whole bunch of tools. And the key thing is, I think, to keep track of the versioning of these tools and the provenance of how the analysis results were generated using what tool and so on. So for that, again, Flyte is perfectly suited. So we have a list of how were these results generated? Was it using Minimap 2? What version of Minimap 2 was used, and so on. And we get a nice set of results at the end, which is much easier to digest for biologists. If you go to the next slide, I think is so any questions so far on what we were trying to do? I know some of that is very superficial. If you have any questions on how exactly I've implemented some of these things, please feel free to reach out. We can have a chat about that as well.
I do have a question for you, Krishna. So dashboards is I think a very, very good tool to share results. Is there another need to create? I mean, usually the more scientific, documentation and reports with how do you do this, or is there no such need? Is dashboard exactly what you have to do? And then when you put together a paper, that's all a different manual process. How do you create papers, science papers?
Oh yeah, so that's all a different process. So this is we don't generate reports yet, but in the future, probably, once we go down that route of, if we wanted to, one of the things that we were talking about recently is if you wanted to make a report of all the successful mutations, for example, how do we pull those things? And is there a format for it where they say, when someone goes in there and says, hey, give me all the successful mutations for this particular gene that we tested, can we provide a report of all the things that we tested? When was that tested in the lab, and how did the results look like? And what results were generated for that? I think that would be super helpful.
Yeah, what I have seen is very often the usage of parameterized markdowns, and then you can run things, and then obviously the experimental the job changes, variables change, but the markdown actually picks all up on that and produces the same scientific output.
Oh, interesting. Okay, yeah, that's something we can chat about and see how that's implemented. Okay. Yeah, I've never done that before, so cool. Interesting. Any other questions or comments? Cool. So yeah, so a few things that I loved so far. Super helpful team. I can't say this enough. And everyone's been super responsive on Slack. Examples on the website and deployment walkthroughs, especially when I first started out, were also super helpful. Kubernetes, like I said, was what we were looking for. Integration with AWS was very nice. Shell tasks was important for a lot of bioinformatics tools. Container tasks, obviously, for working with different kind of languages like R and Julia. Map tasks was super nice. I still don't know I'm using them correctly. I need to dive into them a little bit more. The one thing that I really liked compared to, I think, my previous experience with some of these tools was the local dev experience with PyFlyte and the sandbox. are super, super nice to reduce that friction between production and dev environments. So that was really nice for me to see it. I mean, it's not completely zero, but it's much lower than what I thought it would be when I first started. Flyte console is nice. Ability to cache results is good. Task level resources was super helpful because some of these tools are very, very memory intensive. So you being able to say give them a lot of memory compared to other tasks was also super helpful. Go to the next slide. Sticky things more than anything else, I think, because it was my first time with Kubernetes. Half my battles were trying to understand Kubernetes more than anything else. Flyte, there were some minor differences with the prod versus what I was doing locally. Shell tasks and map, I don't know what the recent status is, but when I was developing, I don't think shell task worked with map. But shell tasks are super, super crucial for bioinformatics, and sometimes these are so small that you can like spin up using map multiple of these things. That would be super useful for bioinformatics pipelines.
Hey, Krishna,
Slack channel. Yep.
Sorry, this is Ketan from Union again.
Yeah.
Are you like? Do you have any repro for the shell tasks with map? Because I think that should just work out of the box, and if it doesn't, we can take a look into it. But we would love to learn your use cases because
yeah,
there's also been a lot of updates in map tasks recently in the last 2 releases. So check it out.
Perfect. Yeah, I can send you a couple of tasks that are, I think, used in almost half the bioinformatics pipelines. That might be a good starting point. Yeah. So a few sticky things. Again, nothing major so far. My experience has been, I think, super positive compared to my expectations when I first started out. So thank you for putting this tool together. It saved a lot of us time. So
fantastic. So thanks, Krishna, for your talk. So I would love to still see if there are a few more comments or questions from the Flyte team here as well. So I think we want to learn a little bit more about the use cases. That was one thing. Anything else where, you know, Ketan, Eduardo, you want to chime in and talk a little bit about the future with Flyte and, you know, potentially have feedback for Krishna?
From my end, I think I would love the map tasks solve your problems as they should. It's definitely, you know, don't think twice about filing issues. It's okay, right? Even if there's a misunderstanding, we can definitely solve it over that issue. So, it's all the documentation, right? That's a great part.
Yeah, like I said, part of it is probably me not keeping up with things in the past few weeks. So
but this was great. Like, it really helps us understand the landscape of, you know, we've had a few folks share their journeys of, you know, in biotech recently, and a few more lined up, I think. It's just amazing to hear that there was this tool exists that can solve, you know, problems within a world that it was not designed in the beginning to solve for. Because we are working, we had no idea about this, but it's great, and we want to make it better now for you guys. So please let us know.
Yep. It's definitely, like I said, one of the best tools I've used so far, and the proof, I guess, is how much time it's saving the biologists right now, and they all love it. So I wish I could give you their testimonies, maybe in the future.
Right. Thanks. Krishna, thank you so much. We really appreciate it. Let's go back to the slide deck and to the next part. So really appreciate your insights, Krishna. We will have you back. And once we get to the other parts and make progress and learn about how your workflow evolves, we would like to do that and talk about that. So thank you so much.
No, thank you.